Okay, thanks for the introduction, Sarah. So today I'm going to be talking about my recent preprint and the R package called hdWGCNA. And yeah, so I'm a fifth year graduate student at UC Irvine in the Seurat lab. And we normally study, we use like neurogenomics to study Alzheimer's disease and other disorders. But this is pretty much purely a methods paper. So I will kind of tell you how I arrived at that point when my main focus is actually on, understanding a disease, not really the method development. So just for an outline of the presentation, I'm going to first go through the background and motivation behind this study, and then a brief overview of the hdWGCNA R package, followed by an application in spatial transcriptomics data, and then another use case using single cell long read data to do isoform-level network analysis. And then I would say this is like the most real world application is actually using it to study a disease condition. And then finally, we'll just do conclusions. So first, for some background, I want to talk about, we can't understand what is hdWGCNA without talking about first about WGCNA. It stands for Weighted Gene Co-expression Network Analysis. And this is actually quite an old method. If you look at this paper from BMC Bioinformatics, it was published all the way in 2008 when I was in middle school. So why are we still using this? It's been cited thousands of times at this point. It's kind of a tried and true method for doing network analysis in bulk tissues, whether it's from, it's been used in microarrays, bulk RNA-seq, proteomics, I'm sure other things as well. So it's a very like flexible and generalizable method. And they do have some decent, tutorials to go along with this. This figure from the paper on the right side just kind of goes through the basic workflow. So starting from some, samples by genes or samples by proteins table, you can construct a co-expression network. And for those of us who don't do a lot of network analysis, I guess like when you imagine a network, you probably think of like dots with connections in between them. But the way a computer actually sees a network is just a matrix. So this is a representation of a co-expression network where, so this matrix it's a square matrix where you have, it's a genes by genes square matrix, and the value that is there is going to be essentially how connected they are in the network, or whether or not there is a connection. So it's, for this weighted network, it's not just a 0 or 1 for whether or not there's a connection. There's actually a continuous value that tells you how strong the connection is. And the value is based on correlation of the gene expression. So once we have the network, and you can kind of see this pattern here. So we want to group genes together that are highly co-expressed, and we call these modules. So the way this is done is essentially hierarchically clustering this matrix. And the same group that developed WGCNA developed an algorithm called dynamic tree cut, which will essentially group these things together inside of this hierarchical clustering. And there's a lot of different parameters for dynamic tree cut. So you can get like fewer groups or more groups, or having a minimum size group, or merging groups that are too similar, things like that. And then all of this is kind of like the core part of WGCNA is these two steps. And then everything downstream is kind of like a choose your own adventure. So typically we want to kind of like, we want to relate modules to external information. So like, because it's an entirely unsupervised method, you want to figure out like what biological processes are each module involved in. So you might want to do like go term enrichment. The next thing that's typically done is like module relationships. So since it's a hierarchical clustering, you might want to figure out like which modules are expressed in the same samples, which are expressed in disease, etc. Another thing we can do is find the key drivers in interesting modules. So these in a gene module, we can look for the genes that have the highest connectivity. So you can imagine that if a gene that's highly connected in a network is perturbed by a disease or a drug or something, it might have downstream effects. So we can kind of prioritize key driver genes in that way. And I have been working with WGCNA since the start of my PhD. This is from a paper we published a couple of years ago where we did network analysis in Alzheimer's brain with bulk RNA-seq. So we took all these published bulk RNA-seq data sets from different brain regions, and we wanted to do something called consensus network analysis, where we're constructing a network and where the edges are kind of conserved across all of these different brain regions and these different brain banks. So they're kind of like, we could take a different approach where we're doing different networks for each region, but we wanted to see like what is like a unified co-expression network across all of these. And then down on the left, we wanted to basically correlate the expression of the module to the AD diagnosis. And so we have things that are going up in AD and going down in AD. And then this is just a representation of all of the different modules, and we have like a top go term for each and like how they're correlated with one another. And then following this study in my paper from last year, we had a single cell RNA-seq study of AD. And this is kind of where we started to develop the predecessor to hdWGCNA, which we called scWGCNA. So this is like just a snippet of the results of that paper where we took the oligodendrocytes and then we ran scWGCNA on it. So each of these network plots are going to be the hub genes, the top 25 most connected genes for each of the oligodendrocyte modules. And then we did correlation with AD diagnosis. A different part of this paper, we found an interesting transcription factor. So we wanted to kind of overlap the target genes of that transcription factor with these modules to kind of infer if that transcription factor might be regulating these modules, and so on and so forth. So I think like the way we developed scWGCNA, it was kind of like, it kind of was like a script to get the job done at the time, but it wasn't very like nice to use, I would say. But earlier this year, I was actually starting to see people using it. So I was like, why are people using this? Like, it's kind of awful for even me to use. But this paper that I saw in Nature, they actually did use scWGCNA. But of course, these plots look like very different than the ones I showed here. That's because the original scWGCNA didn't include any plotting scripts. So it kind of left all the plotting up to the user. So this kind of brings me to like the motivation and goals for developing hdWGCNA. So as I was kind of saying, scWGCNA was like merely a bridge between your single cell data and WGCNA, but I wanted to create a more end-to-end analysis framework. And I wanted the implementation to be object-oriented. So that would be kind of, for those who don't know like programming lingo like that, that's kind of like how Seurat works, where you have like a Seurat object and then all of your transformations are on that object, rather than the original WGCNA is kind of like a bunch of like disconnected pieces of data that are floating around. So it's kind of easy to get lost if you're a beginner. And then on that note, I wanted to make like a lot of detailed tutorials to make network analysis more accessible to beginners and to show a lot of different use cases. I also wanted to kind of like make it feel like it was part of Seurat. So all of the functions have the same like naming convention and they work with Seurat objects. I also wanted more built-in functions like for data visualization, for statistical tests, and for gene set enrichment and overlap analysis. Other things, like some people really don't like the colors that WGCNA makes by default. So I made it so you could like change all the colors, lots of different built-in functions. So on that note, I'll start talking about the actual R package. This is kind of the screenshot of the landing page on the documentation website. So it's a lot more than what scWGCNA was, just one README with like a short tutorial. This is the full documentation website that is still being updated. We have, I think this is like 12 or so vignettes. So we have different tutorials for different use cases and for more advanced analysis as well. This is just a figure showing kind of like the basic workflow of going from a single cell data set to having your co-expression network. So the first step is essentially to go from your raw counts matrix, like the result of cell ranger, to like a clustered and annotated object. So this can be done using Seurat or ScanPy or additional external packages. And for full disclosure, I like typically use ScanPy, but this package is in Seurat, or it's in R based on Seurat. So it is interoperable with ScanPy or other things like that. And then up to this point, I haven't really described like what makes this like suitable for single cell, but I will start to talk about that more. One of the things that's like an issue with running WGCNA on single cell data, since the the edge weights between two genes are based on the correlation, we actually have an issue with computing correlation between two genes in single cell due to the data sparsity. For example, like data sparsity, like in single cell comes from, part of it comes from biological signal, part of it is from technical noise. So if you have like two biologically similar cells or groups of cells, two different data sets, and you're correlating two genes in both of those data sets, you actually might get pretty different results just due to how sparse the data is. And what I mean by sparse is like if you have a measurement for all 20,000 or 30,000 genes, how many of those measurements are zero? In single cell data, most of them are zero. So we kind of wanted to do something that would reduce the technical data sparsity while retaining the biological information. And the way that we do that is something called like constructing something called metacells. And we're not the first ones to come up with this metacell concept, but essentially you want to take, it's almost like clustering, but you want to take very, very small groups of cells and put them together. So the way we do this is on a reduction of your choice. So it could be PCA, harmony, whatever reduction. You can apply K nearest neighbors, and then you can basically merge. You can do this like sampling process to merge, have as many neighbors as essentially you want. Typically like 25 or so cells get merged together, and then you can have like a target number of metacells, because we could have essentially like, it becomes like a combinatorial problem, which is like, for some people, they don't understand, like if you have like 5,000 cells, you could have like, the number of combinations of possible metacells are like much higher than that. So you have to kind of constrain this algorithm by having a target number of metacells. And we can also ensure that there's like not too much overlap between the different metacells. So that's something we also account for. And then this is essentially all still stored in your Seurat object, and then it can be used for downstream analysis. And another thing to note about the metacells is that they're actually, typically we do this process separately for each biological sample and each cell cluster. So you're not gonna get a mix of different like brain cell types. You won't have neurons and glia colliding. Also on that note, this whole package is very flexible. So if for some reason you wanted to allow for that, you totally can. So the next thing you want to do is set up the expression matrix for the analysis. So like in this case, if you want to do network analysis just on one cell type, you have to specify, like explicitly specify which cells you're going to do it on. Or if you just want to do like, let's say you want to do like a male versus female network. So you'll be like, I want just my neurons from male samples. And then you can kind of proceed down. And then typically like you would probably go back to this step 3 to do to make another network. If you wanted to like kind of have a different network for each cell type or for different conditions, etc. And even the Seurat object can actually store like multiple different hdWGCNA networks, which is pretty convenient. The next thing that we do is like we do this hyperparameter testing for one parameter. Just one of the most important ones for WGCNA is the soft power threshold. So like I was saying, the co-expression network is based on correlation, but we do this kind of soft thresholding. So it's not just a 0 or 1, like I said, it's a continuous value, but we take the correlation between the two genes to a power beta. And then, so, if we have like a small number taken to a power, it becomes a really small number. And it's like less than zero, or sorry, less than one. But if you have a bigger number taken to a power, it becomes bigger. So after doing this soft thresholding, all the really weak correlations will go to nearly zero. So this is important for kind of reducing the complexity of the network. So this testing will essentially tell you the ideal range of values for the soft power threshold. And if you don't get above the threshold, then you probably need to revisit your previous steps. The next thing that's done is the construct network function, which just, it takes your soft power threshold and it takes your expression matrix that you set up previously, and it computes the co-expression network, and it does the dynamic tree cutting and hierarchical clustering to give you the gene modules. And then the next thing we want to do is kind of compute the gene expression for each of your co-expression modules. And the way that was done in the original WGCNA is by computing something called the module eigengene. So if you have your whole expression matrix like up here, we actually just take the just the genes from one module and then compute principal component analysis on that subset of your gene expression matrix with just those genes. And then the first principal component is called the module eigengene, and it represents the expression of that entire module. And since we, with single cell data, like PCA often is confounded by batch effects, we can actually apply harmony to correct for that. And we can, before even doing the PCA, we can also apply regression to account for any continuous covariates, like percentage of mitochondrial reads or things like that. So there are definitely these new options that are not included in the original WGCNA to account for these very single cell specific things. And the other thing we do is compute it's called the eigengene based connectivity, or KME, which is very simply just the correlation of the gene to the module eigengene. And that gives us a proxy for how connected the gene is in the network. So that'll allow us to identify the most connected genes, which we refer to as hub genes. Okay, so this is kind of just the basic workflow. And then I'll start to go into like the first figure of the paper, which is like, I would say like this is like a simple example of using this. If you have your single cell data set and you want to like iteratively loop through your major cell types to construct a network. So for example, like in this UMAP, this is a data set of the brain. So we have neurons and glia. So starting with the astrocyte population, you would construct your metacells, and then you would set up your matrix that just contains the astrocyte metacells, and then construct the co-expression network, and then go off for all of your different downstream analysis. This plot in panel B is actually showing, this kind of illustrates the point I was trying to make earlier about the gene-gene correlations. So the different lines are basically, the darkest one is for single cell data, and then for different values of K in the K and N. So like this is combining 10 cells into a metacell, 20 cells, 30, and so on. So the distribution of these gene-gene correlations in the single cell data is very much like a spike at 0, where nearly everything is like 0 or like, I don't know, 0.05 or something. And this distribution flattens a lot when you are looking at metacells instead, meaning you have like more correlations that are not just at 0, right? So this makes it like plausible to even do co-expression network analysis in this data. And then panel C is actually showing us the sparsity as if like sparsity versus the metacells. So on the very left, we have the single cell data, and then on the very right, we have pseudo bulk. So that's like pseudo bulk is just combining all of the cells together for that cell type. So then you can kind of see that it starts to plateau. Oh, does somebody have a question?
Yes, there's a question. Let me just read that out. So this is from Zechuan Shi. In the middle of A, does that mean metacell can share cells?
Yes, so yes, that's correct. The metacells, yeah, you can see that there's this overlap between these two in this cartoon. and that is intentional. The metacells do have a tolerance to share. You can actually provide that parameter as the user. Like if you say, I want to combine 25 cells together into a metacell, and I only want at most 5 cells to overlap, something like that.
Okay, great. And there's a second question from Edo. Is there any preference in using Pearson over Spearman for the correlation matrix?
So that's a good question. And I think as of now, the package just uses, WGCNA has its own correlation called bi-weighted mid-correlation. And I can't really remember off the top of my head what that means right now. But I do believe that this package does allow you to use like whichever correlation method you prefer. And I just kind of was taking like, for a lot of this, taking the like knowledge of the people who have been doing WGCNA for over a decade, who generally prefer the bi-weighted mid-correlation. So I'm like, okay, that seems like a good thing to use.
And the questions are just coming in now. Really a lot of interest on this slide. So we've got one from Anne Baptiste who's saying, how sensitive are the results to the degree of overlap of metacells?
Yeah, so that one, it wasn't actually included in the paper, but I had an analysis of that. And there is like, if you choose really high overlap, that's not very good. And if you choose very little overlap, I guess it's like how like similar are the results with different numbers of metacells is a good way to think about it. And they are like quite similar, but it might like slightly affect which are the like best soft power threshold or other parameters for downstream steps. But I think overall it's like quite similar. I think like if you have too few, the degree of the overlap. So the other thing I guess that matters is like how many cells do you want to put together. So if you have too few, then the sparsity problem isn't really going to be reduced. So like I think generally we say like 25 is kind of where it starts to plateau in this data set. So like I don't remember what the default value is. I think it's 25. Mm-hmm. But yeah, the degree of overlap, I think like the other thing is like if you say like I want 0 overlap, then you actually might end up with like a very small number of metacells overall. So it's good to have like a small degree of overlap. I think like typically the like, I wouldn't say it's like a rule, but it's kind of like, as I've done this package a bunch of times and used it on different data sets, I found that like you will probably want like at very most, like if K is your number of cells you're putting together, like let's say K is 30, you probably don't want more than 15. So K over 2 should be like the very most overlap. Mm-hmm. And the next question.
The last question from Angelique Golding, and she's saying, is there a specific reason that metacells were used over pseudo-bulking all the cells of a particular cell type of a sample?
Yeah, so I think that like the meta cells versus pseudo-bulking is a good point. And I think that essentially pseudo-bulking doesn't really account for cellular heterogeneity in the same way that metacells does. Because you're kind of merging very disparate cell states. Like in this data set, well, this is just the very coarse grain clusters, but for example, excitatory neurons in single cell data sets typically cluster by a cortical layer for like cortical neurons. So if we put them all together, you might have like a mix of things that you don't really want to be mixed in a pseudo bulk. Although like this package is very flexible and you could totally just do pseudo bulk if that's what you prefer.
Great. Excellent. Thanks.
Okay, so I will just move on. The next part of figure 1 was kind of showing just one of these results, which is on the oligodendrocyte network. So on the left side, we actually have a UMAP of genes. So the way that this is done is taking the co-expression network, which we can think about as that matrix. And then we kind of do like feature selection by just taking the hub genes. So then we have a genes by hub genes matrix, and the user can specify how many hub genes per module. And then you just stick that into UMAP, and then it gives you this. So it's still, unsupervised UMAP, kind of like, you can think about it similar to this, where like genes with similar co-expression patterns should group together. So not surprisingly, the modules kind of come out of this. And I'm not trying to make some like brand claim about what this means based on interpreting the UMAP. I think it's just a nice way to visualize the network. And we can see the edges in between the different genes are the co-expression relationships. And importantly, just for like, so it doesn't overwhelm like the plot, the edges are typically downsampled in this plot, just for like visual clarity. And what's plotted here, I think, are the top 2 hub genes per module on this left side. So then we can look on the right side in panel G at the module eigengene, so the expression of each of the modules. And so this bottom left cluster is going to be the oligodendrocytes. So 8 of the 10 modules are pretty much exclusively highlighting the oligodendrocytes. And then interestingly, one of them is in the neuronal cluster, which sometimes does happen in WGCNA or hdWGCNA. You'll have like, this basically means these cells or these genes are probably lowly expressed in oligodendrocytes, but they are all, they're all co-expressed, so they still show up as a module. But if you look at the entire data set in that context, they're actually probably like neuron genes. This next thing that we do is a statistical test for module preservation. So this is kind of asking the question, okay, I have this co-expression network and these modules from this data set. What if I look at this other data set, like an external validation data set, to see if those modules are reproducible? So the way we look at this is by computing this Z preservation statistic. And there is an entire paper on this topic that is very long and very dense with math. So if you want to understand exactly what Z preservation means, you can feel free to go look at that. But essentially we have these like, this like threshold at the bottom is like under here, it's not preserved at all. Like it's not reproducible. In the middle, it's like, okay, it's a little bit reproducible. And then at the top, it's highly reproducible. So most of these, all of them except for the one that's highlighting in the neurons, are at least slightly reproducible. And we also usually look at it as a function of how many genes are in the module. So this is telling us like, okay, this network, when we look at it in an external data set, all the modules that we found that are oligodendrocyte specific are reproducible. So that's a good sign. And then I was also interested to see, like, aside from our own metacell approach, does this work with other options? So there are 2 other algorithms out there, and there's probably more, but this one called metacell 2 and this one called C cells. So we took, we're actually looking at a different data set now, which is, this one comes from the C cells paper. And I did that because I found this algorithm to be very slow. It didn't actually run in any timely manner on this data set, which is, this one is like 30,000 cells. This one is like a couple thousand, only one sample. So for that reason, we're looking at this data set, which is like bone marrow progenitors, I think. So we basically ran the hdWGCNA pipeline the exact same way for for each of these, except for swapping out the metacell object. So for these 2, I just followed their recommended tutorial to construct the metacells. And then, so in the middle here, we're looking at the dendrogram, which is showing us like the hierarchical clustering of the genes in the network. And then we show down here the different modules, and then like below the modules, like how are these genes grouped from the other 2 methods. And then on the right, we have the module eigengenes. And like, I think this isn't like really like benchmarking or anything. This is just kind of telling me, okay, we get like suitable / reasonable results using these different metacell approaches. They are all slightly different. And like, one thing that I found interesting is that I don't think either of these actually account for the biological sample or the cell type / cell state. So like, this means like cells from, if you're interested in like Alzheimer's disease, you probably have like case and control. So this would mean like cells from case and control could actually, like by following their default tutorial, they could actually mix together. So you'd have to like write your own custom script to get it to work in the way that you wanted it to if you didn't want to mix up those things. And yeah, even doing this, like I would say this is a very like advanced or special use case of hdWGCNA. We do have a whole tutorial. If you really like C cells or MC2, you can totally use that in place of our metacell algorithm. So that kind of covers the very basics. And now I'm going to show an application in spatial transcriptomics. So for those who aren't maybe familiar with spatial transcriptomics, it's exactly what it sounds like. Oh, wait, so we have a question.
Yeah, there is a quick question from Melissa Salazar. Hi, quick Q. Although you mentioned in the paper that metacell methods are comparable, would you recommend one of these methods in specific cases or data sets?
I would just recommend the one that is included with hdWGCNA out of convenience, because I can't see a clear advantage in any of them. Although, like I said, the C cells method in particular was really slow, like prohibitively slow, I would say. Whereas, like, I don't show any of this in this presentation, but we did try hdWGCNA on a 1 million cell data set, and it worked in a reasonable time frame for having that much data. So I don't even know how long I'd have to wait for C cells to get through 1 million cells.
Okay, great, thanks.
Yeah, so spatial transcriptomics is kind of like when you have a tissue and you kind of want to see like exactly what gene or protein is being expressed in a certain cell at a certain location. That's kind of like the goal the field is striving for, but at this moment, it's not. There's kind of like trade-offs and drawbacks for different technologies. So the one I'm going to talk today about is Visium, which is from Neurogenomics. So you can see here, it has like, I don't know, around 10,000 barcodes versus like you get like all the genes. So like the full transcriptome, as compared to like other methods like MERFISH, you get much fewer genes, but you get, like a lot more cells. So that's kind of the trade-off. And it's important to note Visium is it's not a true single cell method. You have essentially like a grid that covers your tissue sample, and anything in this spot at that like point in the grid is going to get a barcode. So that could be multiple cells. And then, for neurons that might be like large cells, they could actually, one cell could be in more than one spot. So it's like definitely a computational nightmare in some ways. But this data set is still very interesting. And this is just kind of one that's publicly available from 10x Genomics. This is also the one that's like on the Seurat website. So we just downloaded it from the Seurat website and clustered it using that tutorial. And then we kind of get, we like cross-referenced it with we looked at the gene expression of the marker genes, and we also looked at the anatomical region where this cluster was. And then looking at the like Allen Brain Atlas, we could give it these annotations. So then we wanted to plug it into hdWGCNA to see what co-expression modules there might be in here. So instead of doing like meta cells, we have an alternative algorithm called like meta spots. So starting from this tissue sample, we can kind of place this grid down of principal spots. And so this is just like an evenly spaced grid throughout the tissue. And then essentially for each principal spot, we're going to merge together all of its physically proximal neighbors. So then if you think about like, okay, how much overlap is there in between two spots? It's at most, I think like 3. Maybe I'm counting wrong. I don't know. It's not that much of an overlap, but this one actually ultimately has fewer parameters because we don't allow you to do like bigger or smaller meta spots at this point. It's just this. So you're grouping 7 into 1. And this does reduce the sparsity. And we kind of see the same flattening of this gene-gene correlation distribution. So we thought this was like a suitable approach to move forward. And then this is looking at the co-expression network UMAP and the module eigengenes for the spatial network. And so we can kind of see like broadly by looking at the module eigengenes, there are different expression patterns for the different modules. And I will say that while in the single cell one, we would do an iterative approach for different cell types. Here, we just did like a whole brain spatial co-expression network. Oh, whoops. But in the supplementary, we do this just on the cortex. So you could do like, you could do different, like you could kind of do it iteratively like that. But if you have like too thin of a section, like you're like, I want to do just one cortical layer, then I think it's actually a limitation of this algorithm where you can't actually group things together because there might not even be like 3 up or down, like adjacent spots, if that makes sense. So here we're looking at we're looking at each dot being a gene, and we can highlight the hub genes. So like we see some myelination genes in SM1, which is going to be highlighted in the white matter tracks. And we highlighted some of the Go terms for a couple of these modules. So like this SM4 module, which is highlighting in the blood vessels, is enriched for terms like blood-brain barrier, and so on and so forth. So this is kind of giving us some context to what these genes and these modules are, and where they're expressed spatially. So we do have this tutorial to give you the entire workflow of working with this spatial transcriptomics data in hdWGCNA. At this point in time, just for Visium, if we do have any interest, I would definitely be able to apply this to other spatial transcriptomics technologies as well. And then the next part of this talk, I wanted to go over our long-read single-cell analysis to do isoform co-expression networks. So for those who might not know, long-read RNA-seq can give us like isoform-level resolution, whereas short-read gives us just like a readout for that gene. But we know biologically, different isoforms can lead to very different, protein products and ultimately downstream function. And then there are recently been developments to kind of merge single cell and long read. So 2 of these papers came out in the past year or so. This method is called scissor-seq from the Tilgner lab. And then this method is called LR split-seq, which is from our neighbors at UCI, literally the lab down below us. So we worked with this person, Fairleigh Reese, is the second author on the hdWGCNA paper. So she was, we were talking about long read and I was talking about WGCNA, and she's like, yo, you should try to run that on this single cell long read data. So I was like, cool, let's do it. We actually use the data set from this paper because all of the examples in the hdWGCNA paper are kind of like neurogenomics related, whereas their LR split-seq they did on muscle cells. So it was more appropriate, I think, to use this data set. Oh, we do have a question.
Yeah, there was a quick question, which was on the metaspots. So what is your opinion on the fact that the spots are a mix of multiple cells? How do you interpret the annotation of these modules?
Yeah, so I think the thing is with these the spatial modules, this is like telling you more about like what's going on in that region rather than like what cell is expressing it. You can kind of infer, like we know oligodendrocytes express MOBP and they're enriched in the white matter tracks. So we can infer that's being expressed by an oligodendrocyte. But the brain has like a lot of different cell types. And for some things that are like less common, like microglia, they're kind of spread across the whole brain. And there's like relatively few compared to the neurons and the astrocytes and other glia. So at least in my experience with spatial transcriptomics, it's hard to pick up on the rare cell types. But so yeah, overall for the interpretation, this is just mostly telling us like, it's definitively telling us like, where is it expressed? And we can kind of like take another step to say like, who might be expressing this? And we can cross-check with single cell data to see like, oh, I have these 25 hub genes, which one's expressed by which cell type in this like complementary data set. But I think Visium on its own is definitely hard to interpret. Hope that answers your question. So yeah, for the long read single cell stuff, we are using this data set from the scissor-seq method, which is essentially just I think they've improved upon this method in a second paper that was published this year, but this one, they essentially just took their single cell suspension, they did 10x barcoding, and then they kind of take separate aliquots for short read and long read. So you do have like kind of multi-omic readout for some of the cells where you have like short read and long read data. So the long read data is like, I don't know if anyone here has worked with like single cell ATAC-seq, but there's even more data sparsity. So it leads to more, like, it's more challenging to cluster your data and to get like accurate cell type labels. So at least for this data, it's nice to have the short read to be able to cluster. But then once you have those clusters, you can look at the different isoforms that are specific to different cell types. So we took this, just the hippocampus data set, and then we reanalyzed it just using the most basic Seurat pipeline. And then we used, I don't think these are the exact same labels they had, but they're very similar. And I wanted to kind of just take one group of cells, so this like radial glia lineage. So radial glia being like progenitor cells that are still present in the hippocampus at postnatal day 7 in the mouse. And they give rise to all sorts of different cell types, including neurons, glia, astrocytes, ependymal cells. I think they definitely give rise to oligodendrocytes, but there was like strangely like there's an OPC population here, but there was like 0 oligodendrocytes in this data set, so whatever. But since it's a developmental lineage, we wanted to kind of perform pseudotime to get a continuous trajectory from the radial glia to the terminal, or like, I guess not terminal because like this one is NPC, but basically to the end of this cell state within this UMAP compartment. So following the pseudotime, we ran WGCNA on the isoform matrix from this data set. And this gives us a similar looking co-expression UMAP to what we've seen before. But if you look closely, like at this module M6, you can see that the hub genes highlighted like are actually isoforms. So it's gene name dash number. And like for this one, 2 of the different hub isoforms are both from the same gene, GFAP. And then here we can see the different Go terms that are associated with it. So like cilium assembly is enriched in module 1. So maybe that's an ependymal module. Some of these are like astrocyte differentiation. So that's probably an astrocyte module. And then we can look at actually the module eigen isoforms as a function of the pseudotime trajectories for the 3 different trajectories. See like how are they changing throughout this developmental time course. So we can see like M1 and M2 are they're kind of like lowly expressed in the radial glia, and then they turn on in the ependymal cells. And then these blue ones are like the astrocyte modules, so like M5. Interestingly, it kind of just like stays there, as well as the other ones drop off. And that's kind of interesting because radial glia are known to be transcriptomically similar to mature astrocytes. And then we can see similar things for the NPCs, so like M8, M9. So this is kind of telling, this tells us something interesting about the development, but this also just shows that in general, we can look at these WGCNA modules with respect to continuous cell states, not just discrete clusters. And then the next thing we noticed is that actually we found it interesting that these different hub genes were showing up in different modules. So like we found that out of these genes that are here, 60% of them only have 1 isoform, and about 18% have multiple isoforms that are all assigned to the same co-expression module, but about 20% of the genes have multiple isoforms that are spread across different modules. So this is potentially very interesting information about the biological function that is totally missed in the short read single cell data. And just to highlight, like,
maybe because I just noticed the GFAP that you mentioned seems to be hub gene in various isoforms in different modules, right? So it's not just present in different modules, but it's actually a hub gene in multiple modules. Yeah.
Yeah, I think it's in M6, M4, and maybe a couple others.
Oh, there's another question about GFAP coming up in the chat from Michael Latke. Seeing GFAP as hub gene, how strong does the hub gene property correlate with expression level as opposed to representing potential regulators?
Yeah, so I think the way you define hub genes can be different depending on like how you compute them, but the way this is computed is I think it is slightly biased towards highly expressed genes, just because it's based on correlating the gene expression to the module eigengene. So a higher expressed gene would just generally have a higher correlation. But like, this is kind of, if you think about like, GFAP has a bunch of different isoforms, and in and of itself, looking at the gene, it might be highly expressed, but I don't actually have a readout of like how highly expressed these individual isoforms are, but it's kind of like splitting it up into like, I think here is like 6 or 7 different isoforms. So I think that like, honestly, like out of the isoform data, like almost nothing is like highly expressed like you would think of it in the short read single cell data. So I don't know, I think it's still pretty interesting. There might be like a more optimized way to do it for this single cell long read data, but this is a very new like data modality essentially. Okay, so this is just a part where I wanted to just like let's just look at what the isoforms look like. So these two that are in M6, you can see that they have like, you know, they're actually very different isoforms in terms of like which exons are included. And then like, you know, you might think of like GFAP 3 and 5. You know, they have some shared features here, but like ultimately they're in different modules. Same story for H3F3B 0 and 5, which are in modules M11 and M1. And then this example down here I thought was really interesting because like looking at the gene model like this, they look super similar, even though they are in 2 different modules. But then if we look at it like from this perspective, where you're modeling the transcript as a splice graph, so each node is a different splicing site with, you know, transcription start site, internal splice site, transcription end site, we can actually see the only thing that differentiates these two is their TSS usage. So this is this was made with a tool from the second author on this paper, Fairley, called Swan, which is a really cool visualization tool for long read data. And you know, since single cell long read is so new, it's like it was cool to work with someone who is like one of the only people that knows how to deal with this data. So big shout out to Fairley for helping me with this part of the paper. And then the next thing is just applying hdWGCNA to study the systems level transcriptomic changes in AD microglia. So for us neurogenomics people, we probably have heard about disease-associated microglia, which comes from this paper from Cell in 2017, where they looked at 5XFAD mice, which is, you know, a mouse model of Alzheimer's disease, at 4 different time points. And then looking at the microglia compartment, they could see, oh, cool, there's like a difference in the cell states that kind of correspond with this time course. And then looking at the genes that are expressed in this later stage, they found that this was like a reactive microglia state that they termed disease-associated. And the field has been kind of very fixated on this disease-associated microglia concept or like adjacent concepts for a while. So in my previous paper from last year, we had a multi-omic data set of single-cell ATAC-seq and RNA-seq, and we looked at these damn genes in that data set, and we looked at the enhancers that are turning on and off and the genes that are turning on and off with regard to this pseudotime trajectory from homeostatic to DAM. And this is definitely interesting, but we wanted to take this one step further and look at, okay, if we look at this from the hdWGCNA perspective, what do we see? Like, how are the modules changing throughout this trajectory? So we took 3 published data sets of human AD, our data set, and then the Mathis et al. Data set, and the Zhou et al. Data set from Washington. And then we put them all together and integrated them using Harmony. And then we just took the microglia population and performed pseudotime analysis. I'm not showing it here, but we basically found that we oriented the start and the end versus where classical homeostatic markers are expressed versus where the DAM markers are expressed. And then we took all of this for consensus co-expression analysis. So this is kind of an interesting way of doing network analysis, where if you have different sources of data, like these essentially different batches, the edges that are kept in, like, so you basically compute the network for each data set separately, and then you kind of overlap them by keeping the edges that are common. So that is giving you a consensus view, and it's a good way to kind of deal with this multiple data set problem. But you could also use it to, you could use consensus networks to kind of give you like more biological insight as well. Like if you're like, if you have male and female and you're like, what is common? Then you can do consensus network analysis if you wanted to like specifically study that. So the result of this kind of looks similar to what we've seen throughout this presentation. We have this module UMAP. We have 4 different modules here. And by looking at the hub genes and the Go terms, we can see that MGM2 is likely a homeostatic module where we have genes like P2RY12, CX3CR1, CSF1R, and then the Go terms are corresponding to like pretty typical microglia behavior like synapse organization, cell migration, etc. And then the M1 module looks like what we would think of as our DAM module as we think about it from that original Cell paper, with some of the genes they highlighted like ApoE, B2M, TyroBP. The top Go term was microglial cell activation, amyloid fibril formation, regulation of blood-brain barrier. The other one that we found to be interesting was potentially this MGM4 as kind of like an alternative activation module with Go terms like phagocytosis, myeloid cell differentiation, neutrophils, cytokines, etc. So we thought this one was interesting, but this and kind of like complementary to this MGM1. So we can kind of look at the eigengene trends throughout the pseudotime trajectory, and we see kind of like a plateau and then like decrease in expression as we go from homeostatic to DAM for this homeostatic population. M3, we kind of see it as like plateau and stay there. MGM4 and MGM1, we kind of see like a nearly linear trajectory from homeostatic to DAM. So these are likely are like some of the genes driving this homeostatic to DAM transition. Question.
Is a quick question from Anjali again. How large were the modules in the numbers of genes assigned, and how did you decide on the size? So I guess this is specifically for these 4 microglia modules.
I don't remember how many genes are in there, but I like almost always use a minimum module size of 50 genes, so there can't be a module smaller than 50. And I don't know, I would like guess this is like maybe 100 genes per module. The M3 one might have fewer, but yeah, you can mess with the dynamic tree cut parameters a lot to give you fewer or more modules. I think like 4 might be it might be like a low number of modules compared to what you would expect. With the consensus network analysis, you might end up with fewer modules than doing it separately as well. But yeah, I don't have the number right off the top of my head. And ultimately, the size is just kind of determined by the algorithm, the dynamic tree cut algorithm. Anyways, to move on, we looked at we did like differential eigengene expression. So this is a statistical test that is just part of hdWGCNA. So we compared AD and control microglia in 10 evenly spaced windows throughout the pseudotime trajectory. So that's kind of what this lollipop plot is showing, where it's the fold change on the y-axis, and then the x-axis is the pseudotime windows. And so like the x, there's an x over the point if it's not significant. So we can see that, like, for example, even in the later stage of the DAM stage, the homeostatic genes are actually higher in AD, whereas like in the actual DAMs, like some of them are higher and some of them are lower. So it's like interesting behavior that is like maybe not seen by just looking at like AD versus control as a whole. So this is like more nuanced behavior. The next thing we looked at was actually, we ran a tool called SCDRS to single cell disease relevance score to compute GWAS enrichment in single cell transcriptomic data. So that's what this UMAP is on the top right, is the SCDRS score. And then we actually can see that the SCDRS score increases throughout the trajectory from homeostatic to DAM. And then down here on the bottom left, we correlated the module eigengene to the SCDRS score for the control and AD microglia separately. So we can see actually the highest correlation with GWAS enrichment is in the MGM4 module, which is interesting since most of what we would normally think of dams is in this MGM1. And then finally, just like for external validation, this is kind of like what I showed in the first figure, but we did this for a bunch of different data sets. So we did it for human and mouse data sets and find that most of the modules are reproducible. Some of them are actually less reproducible in mouse. I could have gone and looked at more mouse data sets, but they were all reproducible in this one down here. And then the M3 and M4 were not reproducible in this 5xFAD data set. So it might actually be that MGM4 is more human specific. Very interesting to me. The very last thing I will show you, since I'm very low on time, is we can use hdWGCNA to project modules from a reference to a query data set. And that is exactly how like this works down here. So, you know, given one data set, one set of modules, can I see like, how are those modules expressed in an alternative data set? And then, you know, we can also do the statistical test to see, is it actually preserved significantly? So we took actually 2 sets of modules from bulk RNA-seq and then projected them into our data set from our paper from last year. So we can see here, like there's all these different modules, and then we can see which cell types they were expressed in. And, you know, you can do this from single cell to single cell, bulk to single cell. You can't do single cell to bulk yet, but maybe we will do that. We also did, like, in this one, we did do the module preservation test with our attack-seq data set. So looking at the gene activity matrix from the attack-seq. So this is a pretty flexible analysis tool as well. There's a whole vignette on how to do this in the R package. Okay, we do have a question.
Yeah, there's a question. Is it possible to pass a nesting variable, so a latent variable, like biological replicate, for the statistical test, like in MAST, or is it every cell tested as independent replicate?
For some reason, I am drawing a blank and I don't remember. But this is the good thing about this is that like this package is under development, so I could totally just go and like change it to add that option if that is something that people are interested in. I want to say that we do allow for latent variables. I should check though, because we definitely should. Anyways, that is essentially all I have to show you today. So in conclusion, hdWGCNA is an R package for co-expression network analysis in high-dimensional transcriptomics data. And it is, you know, entirely technology agnostic. So we showed that it was very easy to adapt it to nascent technologies like long-read single cell. And, you know, if there was, I think single cell proteomics is very like emerging as well. So I think that it could be adapted for that use case. Very importantly, hdWGCNA takes advantage of familiar data structures, namely Seurat objects, which I think will increase the usability of this. Because I think the classic WGCNA is pretty like not friendly to users, whereas something like Seurat, there's a lot of like wet to dry converts that are using Seurat these days, and I think that I had those people in mind very much for this package. And then, you know, hdWGCNA can be used to study your disease of interest, whatever your condition is, and we have lots more info on our manuscript and bioarchive.
Great. Yeah, there is another question from Anne. Once more, have you compared your networks to those generated by other inference methods? For example, the B-line benchmarking framework published a few years ago.
I have not, and like I think people probably sympathize with me when I say this, but there's like more stuff than we wanted to do than we could include, and like we're hoping that the peer reviewers will ask us to do stuff like that. So I definitely want to actually compare to other types of networks, but we have not done that yet.
And then the questions just keep piling in, but actually everyone at the end, I mean, I know we're running out of time, but if Sam is happy to stay on a little bit longer, then that's fine. Also, people can come online and on video and whatever and ask questions themselves as well. But let's take this one from Melissa. So she's commenting back to Edo's previous question. Could a possible solution be to harmonize for a variable rather than regress it out, or use latent variables?
I think Edo was referring to like the differential module eigengene expression. So this is like, I don't think we can actually, so okay, like the module eigengene can be harmonized. Like this, in this case, we have, let me go back. We have these 3 data sets. So we did harmonize over them. And then when we run this test, the actual like value, like we're doing essentially differential gene expression, but it's not gene expression, it's module eigengene. And the module eigengene has been harmonized. So I think that's like already been accounted for essentially. So I don't know if we want to like double dip by accounting for it like twice, but like it's essentially like up to the user what they think is the best thing to do.
Yeah,
if that makes sense. So both are options, yeah. And then I have an acknowledgement slide just to say thanks to my lab mates and my co-authors, Emily, Nagin, and Fairley, and my PI Vivek, and definitely a big thank you to all the early users, because I don't know, the package definitely took shape when I got feedback from people who were using it. So I really appreciate them.